The overall goal of this procedure is to rapidly generate gene-deficient mice with the help of designer tail nucleases, or TALENs. This is accomplished by first designing and assembling DNA constructs encoding specific TALEN pairs, which serve as templates for in vitro transcription to generate TALEN mRNAs. The next steps of the procedure are to isolate fertilized mouse oocytes and then microinject them with the TALEN. The final step is to surgically transfer the injected oocytes into the pseudopregnant foster mothers. The resulting F0 progeny frequently show site-specific deletions of genomic sequences that often lead to the loss of target gene function.
Although the method presented here can provide insight into gene function in mice, the basic approach can also be adapted to other model organisms such as rats and zebrafish.
First, access the TAL effector nucleotide targeter website. On the site, choose the TALEN targeter program and paste in the sequence of the target gene. For this protocol, use the PCAG T7 TALEN expression constructs and the Golden Gate TALEN and TAL effector kit that are available on AdGene. If other constructs or assembly kits are used for TALEN assembly, settings might be different. For the optimal spacer length and numbers of tail RVDs, choose the Miller et al.